Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Hafizah Muhammad Rosdi 139180. My final year project title was Gastrointestinal Helminth Parasite Infection of Domestic Duck in Penang. Anas Platyrincos domesticus. Dr. Farah Hazika is my supervisor for this project. For background study, the domestic duck was bred for meat, eggs and kept for artistic merit, or we call that as pets. The decreasing productivity of livestock are the factor of economic losses that can reach tens of billions of dollars worldwide due to livestock parasite infection. Ducks are easily got infected by various species of helminth through feeding or utilization of secondary hosts which harbor the larval stage parasite because they are free-range animals. It also affects the duck fecundity. The symptom of tame duck with parasite appear to be retarded growth, lessened number of egg production, decreasing in weight and serious hemoglobin depression. They inhibits in growth, decreasing in weight gain, reduce in egg production, losses of the bowel, intestinal blockage and stress-related failure is the vital problem in poultry production which causes great loss in economic industry. The objective of this research were to determine the prevalence and level of gastrointestinal parasite infection of domestic duck in Penang and the association between gastrointestinal parasite infection with the potential risk factors such as sex and age. The duck breed is the white Pekin, which is an American breed of domestic duck. They obtain food from plant and animal life in and around the pond, but supplemental feeding was also being given. They feed on a range of substratum which likely exposed to the gastrointestinal helminth parasite. The inclination of duck production caused by helminth leads to a wide variety of diseases in ducks. This is the gastrointestinal tract of duck. The food they eat is moved into the esophagus, then they travel to proventriculus. Gizzard was connected to the proventriculus where it also leads into the duodenum, which is part of the small intestine. The small intestine, which was connected to the large intestine. Rectum was connected from the large intestine to the colon. Ceca are the final section where the waste product was stored. Thus, the waste product of the duck was excreted from ceca. Firstly, the parasite infection in relation to their age. The highest density of helminth was found in older ducks, followed by adult and young ducks. The loss of body resistance in advanced age in older group of duck might be the reason they have higher density of helminth. In addition, for their sex, the female ducks record the higher mean density of helminth parasite than male ducks. The female ducks may be lack in immune status to combat the parasite infection, including some hormonal may be associated with this due to laying eggs without getting proper household and balanced nutritional supply. The study site was located at Penang, Malaysia on the northwest coast of Peninsular Malaysia, which are at Bukit Mertajam and Balik Pulau. It is the industrial area where a duck processing plant is operating and the rural area where there was backyard poultry. Due to COVID-19 crisis, the sample collected was only 30 samples in two different places mentioned in table. The simple random sampling method was used in this study. A total number of 30 domestic duck samples with 10 samples were collected in from a free-range duck farming. Meanwhile, another 20 samples were collected from commercial integrated duck farming system. For the commercial integrated duck farming system, the company only provide the gastrointestinal organs. They fed the duck with the pellet 125 grams per duck for the duck to get the exact same size and weight for each duck. The maintenance was excellent where they gave the proper cleaning of the drinking system, feeders and flooring of the facility to maintain in ideal for prevention growth of the pathogen to maintain the quality of the duck. The age of the duck was 2 to 3 months old. For the free-range farming duck, the animal may wander around the backyard during daytime and come back into the backyard in the evening. They were fed with kitchen waste such as leftover rice and noodles, food remnants, vegetation such as water spinach, Besides, the owner also fed the animal with used microbe-activated solution UMAS. The duck are generally medium in size with a body weight of 2.2 kg above. The age of the duck was mostly 6 to 7 months old. The slaughter process, including the feather of the duck, was made by the butcher. The duck was butchered to get the gastrointestinal tract separated into different regions, which is proventriculus, gizzard, small intestine, cecum, and rectum. The gastrointestinal tract of the duck was placed in sterile plastic container having 70% ethanol until further parasitological examination. The whole gastrointestinal tract of duck was spread in a tray and separated into 7 sections, which is the esophagus to crop, the proventriculus, the gizzard, the duodenum, the small intestine, and lastly the ceca. By using fine scissors, each segment was opened and placed into many different petri dishes containing saline solution. Distilled water was used to wash the content through a sieve. The embedded worm in the mucosal layer of the crop, proventriculus, and gizzard were collected by scraping the mucosa. Finally, the helminths were collected, recorded, and stored in 70% ethanol. Identification of helminth was made by clearing the helminth in ethanol with an addition of a few drops of lactophenol and was examined under a compound microscope. The morphological character of the helminth was used to identify the helminth by Sosby 1968. The data was calculated using descriptive statistics and IBM statistic 26. A total of 30 gastrointestinal tract of duck were examined for helminth parasite. Among them, 20 gastrointestinal tract of duck were obtained from Meng Seng Enterprise and 10 gastrointestinal tract of duck were from Uncle Aziz's backyard. It was found that none of the gastrointestinal tract of duck was infected with the helminth parasite in Meng Seng Enterprise. In contrast to the gastrointestinal tract of duck from the free-range ducks obtained from the villages, with 30% duck were infected with helminth parasite. There was only one group of the helminth found in the gastrointestinal tract of all the duck examined, which was identified as nematodes. The nematode species recovered was Capillaria sp. The predilection sites of this nematode were ceca and small intestine. The free-range duck have higher prevalence than the duck from the caged-rear ducks. In addition, the differences are significant, p equal to 0.10, p more than 0.05. Based on their group of cells, there was 70% of male negative of helminth parasite, whereas 30% of female was positive for helminth parasite infection. Female ducks have a high prevalence of helminth infection. Thus, there are significant differences, p equal to 0.10, p more than 0.05 in prevalence of parasite for both sex correlation to the infestation of helminth parasite. It was found that ducks with the age of 6 to 7 months old have higher prevalence of helminth parasite with 30% infection as compared to 2-3 months old ducks with 70% with negative helminth parasite infection at all. However, the association between helminth parasite infection and the age of the duck was significant too, p equal to 0.10, p more than 0.05. The identification of Capillaria sp. by Solbys 1968. The results indicate that the environmental conditions of these two places contribute to differences in number of ducks that are infected by parasite. Free-range duck record the high prevalence of helminth parasite infection, 30%, due to the semi-intensive management of breeding the duck. The egg, oocysts, larvae, and segment of helminth parasite were easily manifested in the duck in this system through contamination from the soil. The overabundance of duck in their habitat, including contamination of food and water, prevent the greening, cleaning, and disinfection. The unsystematic disposal of fecal, which may contain egg, oocysts, and larvae, with the help of requisite rainfall and moist favorable temperature, which are released to the environment as the result, high prevalence of helminth infection. free-range duck which wander around the backyard during daytime and come back into the backyard in the evening. The free-range duck are easily infected by various species of parasite through the ingestion of contamination of food due to their exposure to the wide natural environment. Low prevalence of Helminth parasite infection 0% in cage real duck because they apply the intensive management system in their farm. The sufficient care in terms of quality housing with the dehelminthization programs routing, frequent use of antiprotozoal truss and avoidance of defilement by taking a proper care of the hygiene in duck farm and the systematic disposal of duck feces. There are significant differences between male and female due to female laying eggs and their eating habit. It causes them to easily get the high prevalence of Helminth infection in female ducks. There are also significant differences between male and female association with the Helminth parasite infection despite that both sexes consume the same intermediate host. Nevertheless, their distinct food habits and feeding behavior give rise to high prevalence of Helminth parasite infection in female ducks. Female ducks feed on an ample portion of invertebrate in their diet and they feed more than twice a time than male ducks. Consequently, their possibility of ingesting infected intermediate hosts was higher than male ducks. Analysis of the number of ducks being infected by Helminth parasite relative to the age of the duck disclosed that there are significant differences among ducks which are 2 to 3 months old and the ducks which are 6 to 7 months old. An older group of ducks record a high infestation of parasite due to loss of body resistance in advanced age. The free-ranging system and unsecured management may be the factor of higher prevalence of Helminth in adult ducks. The young ducks normally kept in capacities to keep themselves from harm, injury and especially from predators. As a consequence, less chances of the young duck to be manifested by the source of infection such as diverse parasite intermediate hosts. Only one species of Helminth was found in this study, which is from phylum Nematoda genus Capillaria. E is Capillaria sp. Ingestion of the embryonated eggs from contamination of food and water causes the infection of the Capillaria sp. Their intermediate hosts have various species of earthworm, Lumbricina. For the conclusion, ducks reared under a free-range system happen to have the infection of gastrointestinal parasite due to the scavenging behavior, making them prone to the infected eggs or larvae which are acquired by eating contaminated food in the environment. Thus, the prevalence of Helminth parasite infection can be controlled by having proper rearing system as well as good management practices. The Helminth parasite infection in free-range duck also need to have an appropriate deworming schedule which can be treated best with ivermectin.